Hey, wonderful AP Bio 2 kiddos. This is Miss Lynham, and this is going to be our fourth video on Unit 3, Cell Structure and Function. So we're going to pick up where we left off. And if I remember correctly, we should be starting with slide number 21 on cytoskeleton. Okay, so we're going to go ahead and get started. Cytoskeleton, first of all, if you look at the word cytoskeleton, we can use our root words on this, and you can break it up into cell framework. So we're going to do a little bit of defining here. Cyto means cell, skeleton means framework, and so this is the framework within the cell that's going to help provide structural support. Now you are going to find this in both plant and animal cells. Okay, so we're going to go ahead and put up here eukaryotic cells are going to have cytoskeleton, but if you look down to this little arrow, it is definitely more extensive in an animal cell than it is in a plant cell. And if you think about the reason why, it's because plant cells have a cell wall, which they also use for support and structure, whereas animal cells do not have that outer boundary around the cell membrane. And so animal cells are going to need a more extensive cell framework or cytoskeleton compared to a plant cell. Okay, the other function, though, besides just being internal support, look at the second arrow where it shows you that also cytoskeleton can allow for movement. Because if you remember correctly when we covered kingdom protista, there are little cilia and flagella. Okay, cilia and flagella, I'm going to write right here. Cilia and flagella, which the cilia were the hair-like structures, and then the flagella were the long tail whip-like structures. Both of those are composed of the same material that we're going to find inside of the cell, but they're a little bit different, and those are going to be allowing for movement. So if you look at the function, cytoskeleton provides structure and support. It also allows for movement in the case of cilia and flagella. Okay, another function down here is they're also providing an attachment site for organelles and enzymes, because inside the cytosol, remember the cytosol is the cell gel, but some of those organelles are going to need something to attach to, so that's what the cytoskeleton allows for. Okay, we said it's more extensive in an animal cell, and then there are three types of proteins that make up the cytoskeleton, because if you want to say, well, what does it look like? Here's a picture. There are microtubules, there are microfilaments, and then there are these things called intermediate filaments. So the three types of proteins that make up the cytoskeleton are microtubules, microfilaments, and intermediate filaments. And if you look at the size, you can see why the names are what they are. If it's a microtubule, it looks more like a straw. Okay, it's got a hollow part in the middle. If it's a microfilament, actin is one of the most common microfilaments, and this one is used in muscle contraction. You can see that this is thinner, looks more like strands of hair, and then the intermediate is in between the two, and so it's still tubular, but not as big of a tube as a microtubule. So microtubules are the biggest, intermediate filaments are the next size, and then actin filaments or microfilaments are the smallest. Okay, now I'm going to give you some different names of these, but you can see keratin. You probably have heard of this protein before because it's actually found in fingernails and in hair, but keratin is a protein that is an intermediate filament type of protein, so it fits into this category. So let's look a little bit more extensively. Let's examine the next slide and look at a picture of where you could see this. I love this picture because it's three-dimensional, and you can see how the microtubules, like this blue one right here, see how it's a bigger, fat tubule? You can see them, they kind of go through the cytosol, and you can see in the picture it's three-dimensional, so this would be a mitochondria right here. And then little ribosomes, you can see the little dots right here, they would be connected by the longer kind of toothpick-looking things right here, which are the microfilaments. So you can see it's kind of cool where you have a microfilament attaching to ribosomes to help stabilize them and keep them in the cytosol. And then microtubules, and then you would have intermediate filaments in there as well. So it's a great structural component on the inside of the cytosol that helps to create that structure and support. Okay, on the next slide, this is kind of breaking down the different types. So I'm going to go over the microtubules, microfilaments, and intermediate filaments in a little bit more detail. So you need to get your pen out, and I don't know if there's enough room for you to write here, or if you have a highlighter, we're going to highlight a few things on this. Okay, here we go. We're going to go into a little bit more detail on the microtubules, microfilaments, and intermediate filaments. You do need to know these for the test. I am going to ask you specific questions on the difference between the three and how they play a role in their functions. So if you look down on the slide right here, it gives you a little bit about the structure. This is kind of a chart, so it breaks down You can look at the structure of each of these. You can look at the diameter, the protein subunits, and the main functions. I'm not really going to ask you about the diameter section, but you can see the differences in sizes with them all being in nanometers. And so with the microtubules, they're 25 to 15 nanometers. And then, of course, the smallest are the microfilaments with 7 nanometers. So that's just giving you an average size if you were using an electron microscope on the difference between microtubules, microfilaments, and intermediate filaments. Okay, the structure I do want you to know, the microtubules, they are hollow. Okay, so if you have a highlighter, you might want to highlight hollow tubules right here. And the wall consists of different columns of tubulin molecules. And so go ahead and highlight, this is the protein subunit. Tubulin is a type of protein that makes up these hollow tubes, these microtubules. Okay, and there's an alpha and a beta. If you remember alpha helix and beta pleated sheet, that gives you an idea of what structure that they're in. Remember, secondary structure of a protein. So this is a secondary structure of protein that helps microtubules, forms that hollow shape. Now, as far as the function, okay, like what the microtubules do, you're going to see, if you have your highlighter, highlight maintaining cell shape. That's one of the main functions of microtubules. So they're going to be compression resistant. We kind of call them girders. If you think about girders in a house, if you were building a framework, they're going to maintain the shape of the cell, but we're also going to find that microtubules are what make up cilia and flagella. So when you see a protist that has a flagella, or you see a paramecium that has cilia, those are made up of microtubules. It's part of the cytoskeleton. Okay, don't forget the big word, cytoskeleton, but it's used for movement. Okay, one thing that we're going to see in us, put a star by this, when we get to the unit where we talk about microtubules making up the spindle fibers. Do you guys remember that when we talked about spindle? Okay, spindle fibers help move the chromosomes to opposite poles of the cell during mitosis. And so spindle fibers are microtubules, so they are actually part of the cytoskeleton. And then also to move organelles, because for a cell to divide, we have to split organelles between the two cells as well. So that would all be functioning of the microtubules, whereas in the second column, microfilaments, okay, these are not hollow, they're intertwined strands of actin. Okay, so actin is a huge component of microfilaments. It's much smaller. The function is also to maintain cell shape, okay, but it's more of tension-bearing elements. Okay, the changes in cell shape, you might have a little slight change in cell shape. That's going to be the function of these microfilaments. Okay, put a star by the muscle contraction, because when we get to the human body and we start talking about the fundamental unit of muscle contraction, which is called a sarcomere, we're going to hit on these microfilaments that are made up of actin. And then cell motility, now don't be confused with cell motility here. We also said over here, but this cell motility with a microtubule is cilia and flagella. This cell motility is pseudopodia, which would be in an amoeba, okay, in an amoeba. So if you look at those protists, if it's a paramecium or euglena, it's over here because it has cilia and flagella, that would be microtubule. If it's an amoeba, it has false feet or pseudopodia, that would be microfilaments. Okay, and then let's not forget this star right here. A microfilament functions in cell division, but not for moving the chromosomes and moving the organelles like microtubules do. This time it's to create the cleavage furrow, which if you remember is when the cell pinches in like that and the cell's dividing, to pinch that cell in half to create two new cells. That's called a cleavage furrow, and you're going to need some microfilaments to do that because it pulls on the cell membrane in order to split the cell in two. Okay, and the last category over here, intermediate filaments. Notice the size is in between 7 and 15 to 25 nanometers, so these are in between. These are going to be fibrous proteins that are super coiled into thicker cables. They're thicker than the microfilaments. Okay, and then the importance of these is also maintaining cell shape and then anchorage, put a star by this, to anchor the nucleus and certain other organelles. Okay, and then as well as with the nucleus to form the nuclear lamina. The nuclear lamina is on the inside of the nucleus to kind of give it some structure and shape. That would be this intermediate filament that makes that up. So cytoskeleton is really important. I know that's a lot of specifics, but I do want you to know the difference between a microtubule, microfilament, and intermediate filament, and about how those function in the cell. Okay? All right, moving on to cell boundaries. We're changing the topic now. That's pretty much the last organelle that we're going to cover. Now we're going to get into boundaries. And if you think about the word boundary, It's like a barrier, but in this case, these boundaries are not going to be foolproof. They're going to allow movement of stuff into and out of the cell, because if they wouldn't let stuff in and out of the cell would not be able to do its job appropriately. So the first boundary we're going to talk about is the cell wall. Notice it's found in bacteria, fungi, and plants. It's also in some, we're going to put some protists, but we're going to put not, we'll put a little comma here, in animals. So this is not applicable to us. This is applicable if you were a bacteria. Remember, we even talked about what the cell wall is made up of. If it's in kingdom Archaebacteria, remember it was polysaccharide material. If it was in kingdom Eubacteria, remember it had that peptidoglycan. Okay, we're just readdressing some of that glycan. If it was a cell wall of a fungus, it was made up of chitin. If it was a cell wall of a plant, it's made up of cellulose. So depending on what the kingdom is in the organism that has the cell wall, the makeup of that cell wall is different material depending on what that organism is. But remember, we as animals, kingdom Animalia, do not have a cell wall. So this is not applicable to us. Okay, look at the bullet here. The job of the cell wall, obviously, is to maintain cell shape. Okay, so maintaining the shape of the cell is a function. It's rigid in structure. No matter whether it's cellulose, whether it's chitin, whether it's peptidoglycan or polysaccharide, most of those that are mentioned right there are polysaccharides. The only exception is this one that is peptidoglycan, because it's part protein and part sugar, so it's part polysaccharide, part protein material. But chitin and cellulose are both polysaccharides, just as this polysaccharide that's listed there. Okay, so the job is to serve as a rigid structure, maintaining cell shape, and it also, in the case of osmotic pressure, that we will talk about later, osmotic pressure, it helps to prevent the excess uptake of water, because it helps to create a back pressure that's going to be on that. So if you look in this diagram, here's the cell wall right here. Okay, this is the cell wall of the next cell on this side, and this would be the cell wall of the next cell over here. So you notice there's a little space in between the cell walls. We're going to talk about that, but the cell wall is rigid here to keep that shape, and rigid here, and rigid here to maintain the cell shape of that. Okay, so if we go into more specifics, okay, for a plant cell wall, because obviously we want to focus on them more so than funguses at this point, the plant cell wall does have cellulose, which is that structural polysaccharide. Okay, however, put a little star right here, pectin is a sticky polysaccharide found between the cell walls of adjacent cells. So you would find that, like right in this area, right there, you would have, not us, but they would have this substance in the middle, right in there, known as pectin. So it's just more polysaccharide material found in the area that's named as the middle lamella of a plant. So in that little area would be pectin in here to also get a little bit more structure. Okay, now I want you to know that this cell, if this was cell A right here, and this was cell B, they would need to communicate with each other as far as like making sure that some things could move from this cell to that cell and this cell to that cell. So what you would find are little perforations, little holes in the cell wall that they would have sporadically that would allow for the transport of molecules between cells. Those holes are called plasmodesmata. They're perforations between adjacent cell walls that allow for movement of materials from one cell to another. So in this diagram, you can see plasmodesmata would be right here, so stuff could go from one cell to the next in a plant, which kind of helps them stay continuity between each other. And they can pass stuff like literally cytosol could pass through. They might have ions, some water, different things that the cells would need to maintain homeostasis, okay, or solute molecules, different, besides ions, different solutes, would be able to pass through those plasmodesmata. So the cell wall is permeable. I want to make sure you know that. It is permeable, which means it is going to allow stuff to pass through it. Okay? All right. Okay, the next slide we're going to talk about the ECM. I'm going to abbreviate that for you so that you can get that down. The ECM, it stands for extracellular matrix. You can see it's right here, ECM. Okay, that's the extracellular matrix. So if you look at the word extra, that means outside. Okay, cellular means of the cell. So this is an area, a matrix, an area outside of the cell. And in animal cells, it is quite extensive. Okay, quite extensive. Mainly because we lack a cell wall, we're going to need this extracellular matrix, because look at the function in the first bullet. It holds cells together, protects and supports the cells on the outside. So because we lack a cell wall, animals, that's you and me and every other animal that's in Kingdom Animalia, is going to need a very complex extracellular matrix that will hold the cell together, protect the cell, but also in the second bullet, allows for communication between cells so that one cell can communicate with the next. Okay, now what it's primarily composed of, in this next bullet you can see, primarily these things called glycoproteins. Okay, glycoproteins, if you look at the word glyco, refers to a sugar molecule. Protein tells you it's a protein molecule. So these are proteins with covalent bonded carbohydrate chains that are covalently bonded to those. So it's protein and sugar covalently bonded together, which gives it a little bit more structural support. Okay, that gives it more structure. The most abundant of the glycoproteins that we have in our extracellular matrix is collagen. Okay, collagen forms, I'm going to just write it here, forms strong fibers outside the cell. So collagen, if you look in the diagram, is right here. You can see this is kind of an electron microscope picture view. So the collagen are these big purple strands that you see going across. You can see how extensive they are. They're very strong, and it's on the outside of the cell, because here's the phospholipid bilayer right here, and then here's the cytoplasm, so that's inside the cell. Okay, so this is outside. So you see the collagen fibers going across, but there's more than just collagen. There are other glycoproteins, such as this one called fibronectin. Okay, there's also the proteoglycan complex, which are proteins and sugars that are coming off of collagen, which are extensive like this. So it's kind of like it's a very extensive network outside of the cell. You know, a lot of times we just think, oh, that's just a fluid structure, but you can notice how these things come in and connect two parts of the cell, because this is something protruding from the cell membrane, and the collagen can come across and make an attachment with that, just like this is protruding from the cell membrane. So the attachment site just provides more structure, and it gives it a fluidy environment where this cell could communicate with another cell. So that's the extracellular matrix, and it's quite complex. Okay, the next slide, we're going to talk about some intracellular junctions. So let's look at this word. Intra means inside, okay, or within the cell, and a junction is like a meeting, a joining. So these are going to be also in animal cells, so these are your cells and my cells, and we have different types of junctions between those. Alright, sorry, I had to pause. My dog had to go outside. Anyway, so these are the three intracellular junctions that are in animal cells. Okay, so the first one is called a tight junction. Just kind of look at what tight says, what tight means. Tight means something that's binding, right? So the tight junction presses membranes together very tightly. The benefit of this is it prevents leakage of fluid from the cells. So if we jump over to the diagram, okay, you have to think that over here, I'm going to just draw over here, this is one cell over here, this is another cell right here, so that's the second cell, and then you would have another cell over here in an animal. So if this was in our body, let's say these were skin cells, you would have one skin cell over here next to a second skin cell, next to a third skin cell, so that would be like one after the next after the next. So a tight junction, if you look right in here, you can see there's a little dot from one, and then a dot from another one, and then same thing here, there's a little dot from one and a dot from another one. What these are protein molecules that help to kind of press this cell to that cell so that you don't have excess fluid leaking out of the cell, so it prevents the leakiness of that. This is very common in epithelial tissue. If I have to get my pen to work, I'm going to go ahead and write that here. I'm just going to write it here. It's epithelial tissue, which is tissue found in the skin. That helps us so that we don't lose a lot of fluid, a lot of water out of our skin. Okay, so that's definitely a common one, those tight junctions, prevents leakiness of that, helps us make our skin watertight. Okay, so that's one. The second one are called desmosomes, okay, or desmosomes. These are anchoring junctions that fasten cells together in sheets. It's so that we have one cell attached to the second cell attached to the third cell, which might create a sheet of cells. This is also common in epithelial, okay, epithelial, which is skin tissue as well. And if you jump to the picture, you can see over here, this is kind of a cool thing where the fibers go through, okay, so these are going through, and they're longer, and so you might have several places where you have these desmosomes that helps to fasten the cells together to be in those strong sheets. I am going to write that the protein that's used in this one is keratin. Okay, so keratin is very important. We mentioned it a minute ago with the cytoskeleton, but it's also keratin proteins that anchor this in the cytoplasm. Okay, one thing I did want to mention on this too, because I know I have quite a few athletes in class. If you are a weightlifter and you want to increase bulk in muscles, a lot of you guys know from working out that you can rip, okay, you can rip and tear muscle cells, and when you do that, you get more bulk. This is what you're ripping and tearing. When you rip and tear a muscle cell, you're pulling away the desmosomes or the anchoring junctions between the cells, which then makes them bulkier, and you get that muscle like mass as far as when weightlifters are trying to get bulk up. That's what it is. So they can also attach muscle cells together in a muscle to create that sheet of muscle cells. It's the rupturing of those that creates that bulk. Okay, the third example is called a gap junction. This is very similar to the plasmodesmata. Okay, write this down, plasmodesmata. I can't spell with this. I'm using a mouse to do this. Plasmodesmata. It's equivalent to the plasmodesmata of plant cells. That's what the function of a gap junction is. So if you look down here, you can see in this picture that it's like a channel between the two. The function of that is to allow for movement of cytoplasm from one cell to the next, which is important in communication between cells. So we could actually let stuff like ions, we could let water, we could let cytoplasm. There's a lot of stuff that we would let go through this, small amino acids, other small molecules that are necessary for communication, those would pass through. And so that one, like I said, is very similar to the plasmodesmata in a plant cell. So these are just ways that when you look at how cells are connected to other cells, there are different types of junctions between them. Tight junctions press together tightly, prevent leakage. Desmosomes are going to fasten them to create sheets. That's using keratin. Both of those would be really common in epithelial type of tissue, even though you can have desmosomes and muscle tissue as well. And then the last one is for transport of substances between cells to maintain communication. That's a gap junction. All right, let's move on. Moving right along. Next thing we're going to get into is the cell membrane. We're going to get into the cell membrane.